All right, welcome back. We're in part 4. We're going to move beyond the plasma membrane into the cell here. So the first thing we'll look at is the cytoplasm, because that's really the first thing a molecule would be introduced to once it got inside the cell. So the cytoplasm is pretty simple. It's just really the cellular material outside the nucleus, but inside the plasma membrane or the cell membrane. It's not really a liquid. It's more like a gelatinous mixture thing. It's the best way to describe it. It's not like Jello. It's just kind of hard to find a good example of it, but it's not liquid. It's kind of a half solid, half liquid thing. But it holds and is composed of the cytosol, which is the liquid part, the cytoskeleton, which is the physical structure, which will be in the next slide here, which provides structural support for the cell, made up of proteins, hence skeleton, right? Cytoplasmic inclusions and like other components here in organelles. The cytosol is the fluid part of the cytoplasm. Cytoplasm is just an all-inclusive word for all this on second bullet point. If we're just talking about the liquid component or the fluid portion here, which includes dissolved ions and molecules and proteins suspended in it, then we're talking about the cytosol. So that's really the cytoplasm. Nothing too fancy going on there. Cytoskeleton, on the other hand, is very important. This is kind of like my specialty where I got my research in for my doctorate. But there's 3 different types of the cytoskeleton proteins here, and they all have distinct functions. There are microtubules, which are the largest. Always throws me off sometimes. They form hollow tubes made of a protein called tubulin. They provide scaffolding support for locomotion. They are really important for cell division, separating your chromosomes out. They are also really important in making cilia and flagella. So microtubules are pretty important. Actin filaments, which are the smallest, which are called microfilaments. This one's really utilized for contractility. So muscle cells have lots of actin in them to contract the muscle. They're generally also used for most of the locomotion of the cells in your body, because they are basically fancy ropes that the cell can use to pull on and move itself through its space. And then there are intermediate filaments, which are proteins as well, but they're halfway in size between microtubules and actin filaments. They have special properties that are different between the other two. It's found in the nucleus a lot, but it's also found in nerve cells, to provide them some more elastic or rubber band-like properties. And as well as these intermediate filaments are found at the just on the inside of the plasma membrane of the cell, to provide some structural support there. So there are cytoplasmic inclusions that I referenced before in the previous slide, but they're just lipid droplets, other molecules like glycogen, melanin, and lipochromes, which we're not going to worry too much about here. But you can see a picture here of what the different sizes are, right? So microtubules, 25 nanometers in diameter, intermediate filaments, 10, and microfilaments are 8, or actin. That's what I call this. Now, cilia and flagella are, interesting kind of cytoskeleton components here, but they're really mainly used for movement. Cilia are. There's another type of cilia that we're not going to worry too much about here, but cilia and flagella are really used in this context for movement. They use ATP to move. So really, cilia is more useful for moving materials over the surface of the cell, not moving the cell. They're just like fingers or appendages. So your intestinal tract, your epithelial cells in there, they have lots of cilia. This increases the surface area for the cell to move stuff across it as well as absorb nutrients, right? So that's why cilia are important. And flagella, of course, for humans, the example that we have for that one is sperm cells, highly utilized flagella, because they're like little, boat motors that spin to move through their space. So, here's the question: where would you find these? Maybe try to find examples of where they're found outside. But yeah, there's also microvilli, right? So there's little ones here. Increases the surface area as well. Can be a sensory receptor. They're on almost every single type of cell. They're smaller than the cilia. They don't move. They have actin inside them, again, to increase the surface area. So cilia and microvilli kind of have somewhat overlapping responsibilities, but cilia are way more active in moving things outside its environment than microvilli, which are more useful for like absorbing things and molecules and so forth. Now, centrioles and spindle fibers, these are still made up of microtubules, but these are more, centrioles, I will say, are more localized near the nucleus, and this is where microtubules are really formed from in terms of the cytoskeleton. They help the cell move, of course, because they kind of are a site of growth for the microtubule proteins themselves. But they're also located inside the nucleus, and when they are, they're called spindle fibers, and they're used for cell division. Because they pull apart the chromosomes from one another. Now, that was like the cytoskeleton. Here's some more specific organelles that we'll highlight here. So these are going to be membrane-bound organelles. So they have that phospholipid bilayer forming. Side note, the nucleus has 2 phospholipid bilayers, so it's really like 4 phospholipid layers going on there. So that's that you can kind of see here. The nucleus, obviously mitochondria, phospholipid bilayer plasma membrane has its own, Golgi apparatus, phospholipid, endoplasmic reticulum phospholipid. So all of these are specialized structures that are membrane bound that in order to perform a certain function. We'll start off with the endoplasmic reticulum here. So this organelle is really a network of channel that really almost blended into the nuclear membrane, so they kind of protrude out. They have spaces called cisternae. These are just kind of like small gaps in them, but you can see they're just little channels here. And this is where ribosomes are located. Rough ER typically has the rough ribosomes attached on the outside here. And this is where proteins are synthesized and made. Versus the smooth ER doesn't have any ribosomes, but they help manufacture lipids. They help detoxify because they have other molecules, maybe enzymes, to break down certain substances, as well as store calcium for signaling purposes. So rough ER is where a lot of protein synthesis occurs. Wherever you find a ribosome, protein synthesis is happening. But when we get into gene expression here at the end of this chapter, this will make sense that the rough ER is really close with those ribosomes to the nucleus, because when gene expression occurs, you go from DNA to RNA, which then the RNA has to move outside the cell in order for it to be translated into a protein. And why not just have that translation site right on the outside of the nucleus, right? So here's an actual image in slide B of what that actually looks like. So ribosomes are enzymes, like as I previously mentioned here. They are composed of both the nucleic acid of RNA and proteins. So they are a very unique structure. They have 2 components, a large component and a small component that come together to form that 3D structure that's made up of 2 subunits here. Now, when we're talking about like types of ribosomes, you can have free-floating ones that just synthesize proteins inside the cell, or they can be attached to the endoplasmic reticulum, forming a rough endoplasmic reticulum, to also produce proteins, but these are more specific in terms of proteins that will be integrated into plasma membranes because they'll be packaged and shipped off in the membrane, or proteins to be secreted, again, packaged and shipped off via the Golgi apparatus. Right, so you can have protein synthesis occurs at both locations, both in the rough ER and outside the ER, but where these proteins are being synthesized by these ribosomes, it depends upon what the function of them is. The Golgi apparatus, it's just a flat sac of membrane, right? They also have cisternae, so little channels in the center here. And really, this is just your USPS, your Postal Service, or UPS, or your FedEx for the cell, where it will intake proteins or molecules to be packaged or modified after they've been made, and then secreted for either internal use or external, like exocytosis, right? So, and the Golgi apparatus is really useful for, transport vesicles, right? Exocytosis. So that's why we always like to say the Golgi apparatus is the packaging center, right? Because it's not really synthesizing anything. It's modifying and packaging and then distributing and sending off to specific locations. So here's an example of the workflow here. You have your ribosomes, you have your RNA comes in to your rough ER, right? Protein is synthesized in your rough endoplasmic reticulum. A little protein there. It gets packaged up by the rough ER, which then it gets released to the Golgi apparatus, where it becomes modified and further packaged, and basically the shipping label is stamped on it and then released from the cell, right? So they work hand in hand, they're tandem in terms of protein synthesis and release and shipping, the Golgi apparatus and the rough ER. So I think we'll pause this video here. I think that's been long enough. We'll move into other transport vesicles called lysosomes and peroxisomes in the next video.